This part explains what the whole of genetic engineering reduces to. Genetic engineering comes down to one capability: pulling a single DNA sequence out of an entire genome and making it in bulk. The whole discipline rests on three primitives. Restriction enzymes cut DNA at defined sequences, the molecular scissors. Ligase joins any two ends, the molecular glue. And a host cell copies whatever you hand it. You cut, you paste, then you delegate replication to biology, and the colony grows the clone for you. Nicholl's compression for this is molecular agriculture. You farm micrograms of a sequence rather than synthesize them. Everything after this chapter elaborates those three operations. The generative move is this: a problem of chemical scarcity becomes a problem of biological growth, and growth is free. This part explains what every use of genetic engineering reduces to. Every application collapses to one operation: excise a single DNA sequence from a whole genome and multiply it into micrograms of identical material. That is gene cloning, Nicholl's four-step primitive, amplified through colonies of identical host cells, which is where the word clone comes from. Protein production, transgenic plants and animals, forensic typing, medical diagnosis, each one is the same. Isolate and amplify. Move aimed at a different end. The chapter's own compression is his analogy. Cloning as molecular agriculture, growing large quantities of one chosen sequence. The surprise beneath the field's apparent complexity is that the principle stays simple. Because DNA is treated as a manipulable resource, a substrate you cut and copy rather than decode. Learn the one move, and the downstream applications become predictable. This section introduces molecular biology as the substrate the rest of the book will manipulate. One asymmetry founds everything downstream. A four-letter alphabet, read in triplets, whose sequence is the message carried on a polymer built so each strand dictates its partner. Nicholl's primer exists to hand you that. Structure determines function is the chapter's spine. Complementary base pairing is the mechanism beneath it. A pairs with T, and G pairs with C. From that, the central dogma follows. The one-way copy in which DNA becomes RNA becomes protein. And with it comes the engineer's real prize, hybridization. Because a strand always finds its complement, DNA becomes copyable, cuttable, probable, readable. Every later chapter exploits this single property. The chapter specifies the substrate. The rest of the book manipulates. Read it as the machine's data sheet, its biology only incidental. This part explains the one primitive that underlies everything a genetic engineer will later do: complementarity. A base pairs only with its complement. A pairs with T and G pairs with C. So each strand is the template for the other. And copying, reading, and cutting all collapse to a single lookup rule. That is why information flows in one direction, from DNA to RNA to protein, through a redundant four-letter code. This is the central dogma. Nicholl's refrain, structure determines function, is the whole point of the chapter. Its geometry gives DNA its three defining properties. It is stable, replicable, and mutable. The shape carries the function, and the chemistry is incidental. The engineer manipulates an information system that happens to carry a physical address, never a molecule as bare chemistry. Master the pairing rule, and every downstream tool becomes predictable bookkeeping. This chapter explains how you work with a molecule you cannot see. Everything downstream in the book assumes you can manipulate nanograms of a polymer that is invisible in the tube. The thesis is this: Every method is a transducer that converts an unobservable property into a macroscopic signal by exploiting one physical invariant. Consider 4 of them. The phosphate backbone carries a uniform charge per mass, so length reads out as migration distance. That is electrophoresis. Base complementarity means presence reads out as a bound radioactive probe. That is hybridization. The terminal 3'OH chemistry, poisoned by a dideoxynucleotide, means sequence reads out as a ladder of nested fragments. That is Sanger sequencing. And differential phase partition gives you purity. Pick the invariant, and the quantity or sequence you want falls out as something you can measure. The rest of genetic engineering is composition over these primitives. This section explains the observability primitive that everything downstream rests on. Everything downstream, cloning, PCR, sequencing, presumes you can already handle a molecule you cannot see, weigh, or count directly. This chapter is the book's observability primitive. Its single move is proxy coupling. Bind each invisible quantity to a physical property that already reads out. Concentration becomes absorbance at 260 nanometers. Length becomes electrophoretic mobility because uniform backbone charge fixes the charge to mass ratio, so only size varies. Mere presence becomes fluorescence or radioactive count. Sequence itself becomes a ladder of nested fragments differing by one base. You always measure the proxy you engineered the molecule onto, never the nucleic acid directly. Predict any later technique from this. Find the invariant, couple the unknown to it, and read the invariant instead. This section explains the tools of recombinant DNA and the surprise about where those tools come from. Recombinant DNA reduces to 3 verbs: cut, modify, and join. The quiet surprise is that none of the 3 are invented. Each is a cellular enzyme conscripted for the bench. Restriction endonucleases cut at fixed sequences, a mechanism that begins as bacterial self-defense against foreign DNA. Ligase joins broken backbones, which is the cell's own repair machinery. The modifying enzymes reshape the ends in between. The generative move is complementarity as the composition key. An enzyme that cuts in a staggered way leaves protruding sticky ends. So any 2 fragments severed by the same enzyme carry matching overhangs, and they re-anneal blindly. Cut and join become near-inverse operations, sharing one base pairing lock. Hence the names molecular scissors and molecular glue. This is a test tube algebra, closed under composition, needing no living system until amplification. This part explains why gene manipulation works by borrowing the cell's own tools rather than inventing chemistry. Gene manipulation is a test tube technology with a closed operator set, cut, join, and modify. Every operator is a borrowed enzyme, purified from the cell that evolved it for its own housekeeping. Restriction endonucleases cut because bacteria use them to shred invading DNA. Ligase joins because the cell repairs its own breaks. The generative move is that specificity is addressing. A restriction enzyme reads one short sequence, and its staggered cut leaves cohesive ends that base pair only with fragments cut by the same enzyme. So 2 unrelated pieces of DNA, cut identically, recombine spontaneously. This section explains why cloning, through host cells and vectors, turns a search problem into a growth problem. Isolating one DNA fragment from hundreds of thousands is a needle in a haystack, where the needle is made of the same straw. No chemistry distinguishes the target from the background. The chapter's move is to stop searching and start growing. Splice each fragment into a vector, a small self-replicating piece of DNA, either a plasmid or a phage, carrying an origin of replication, a selectable marker, and one unique cut site. Then drop one vector into each host cell. Now, clonal division does two jobs at once. The first is physical separation. Each colony descends from a single molecule. The second is amplification. Each colony is millions of copies. A search problem becomes a growth problem. The vector plus host pair is the machine of cloning. Everything downstream, selecting which colony you actually want, is deferred to Chapter 8. This section explains why gene cloning delegates molecular sorting to living cells. A single gene hides in a haystack made of the same straw, millions of chemically identical DNA fragments, only one of them wanted. You cannot sort molecules by hand, so you delegate the task to biology. You hand each recombinant fragment to a living cell and let cell division do the work. One founding molecule per cell means clonal growth performs separation and amplification in a single act. Each colony becomes one purified sequence, mass-produced for free. The chapter's primitive is this division of labor. The vector is the minimal passport that makes a fragment legible to the cell, a replication origin, a selectable marker, and one unique cut site. The host is the copying engine. Everything downstream, phage, cosmids, and shuttle vectors, varies the passport's capacity, never the principle. This part explains what a cloning strategy is at the structural level. A cloning strategy is a path. Chapter 6 reframes the tools of chapters 4 and 5. The cutting and joining enzymes and the host and vector systems as edges in a fixed decision tree drawn in figure 6.1. The strategist's job is pathfinding through it. 2 constraints. Fix the root, the experiment's aim, and the source material. Give me those 2, and you can predict the branch yourself. The first and sharpest fork is messenger RNA versus genomic DNA. Messenger RNA pre-filters the genome to what a cell actually expresses, and it has already excised its introns, so it wins for making protein. Genomic DNA is forced only when the target is the non-coding control machinery. No universal strategy exists. Every project is one traversal. This section explains why cloning begins with a single choice, and why that choice does the most work. No cloning strategy is universal. The chapter is a decision tree, and its first branch, begin from messenger RNA or from genomic DNA, already fixes where selection happens. Choose messenger RNA, and the cell has pre-filtered for you only what a given tissue expresses, introns already spliced out, abundant skewed toward what you probably want. Choose genomic DNA and you inherit the entire genome, introns and control regions included, worth the noise only when regulation itself is the target. Everything downstream, the vector, the joining method, the host, is a compromise serving that first commitment. The elegance is that your choice of input, not any later trick, does the heaviest filtering, the cell's own expression. Machinery is your first purification step. This section explains the polymerase chain reaction as a way to amplify a target without a cell. The whole idea is to make the reaction's product its own next template. 2 primers bracket the stretch. You want a heat-stable polymerase extends them. Then you melt the new strands apart and repeat. Each cycle, every copy becomes a template, so the target grows as 2 to the nth power while everything unbracketed stays linear background. That is a millionfold enrichment in an afternoon. The structural primitive is autocatalysis, geometric growth wherever a copy re-enters as template. This is the chapter's pivot in the book's arc. Amplification moves in vitro, replacing the earlier in-cell cloning machinery with nothing but temperature cycling and the right enzyme. This section explains PCR, the polymerase chain reaction, as amplification without a cell. Cloning borrows a bacterium's replication machinery to copy DNA. PCR strips that down to its arithmetic core, a copy step made reiterative. Heat melts the duplex, two flanking primers pin the target's ends, and a thermostable polymerase. Tuck, drawn from a hot spring bacterium, extends them. Repeat, and each new strand becomes template, so copies grow as 2 to the nth power. 30 cycles carry one molecule to about 10 to the 9th. The whole chapter is elaboration of a single mechanism, Mullis's realization that a reaction loop feeds on its own products. Everything downstream, reverse transcription, PCR, nested, inverse, and RAPD is that same loop under one changed boundary condition. It displaces cloning wherever what you actually need is pure amplification rather than a living host. This part explains why selection, screening, and analysis are all one move. You have 1 million recombinants, one wanted sequence, and none of them visible directly. So every method in the chapter does the same thing. It couples the invisible property, carries the gene, to a phenotype you can actually read. There are 2 settings of that coupling. Selection makes the coupling a survival condition, antibiotic resistance or complemented obsertrophy, so the unwanted cells simply die. Screening instead makes it a visible signal, the blue and white X-Gout test, probe hybridization, or antibody stain, read across a whole plate at once. Insertional inactivation is the elegant inversion. The insert breaks a marker, so presence registers as a loss. Everything after identification, the blots, the restriction maps, the sequencing, is the same read at finer resolution. Detection is proxy construction. The whole cracked is binding a readable proxy tightly to an unreadable target. This section explains why identifying one desired gene among 1 million recombinants is the hardest step in cloning, and how the chapter solves it. Finding one desired gene among 1 million indistinguishable recombinants is the hardest step in cloning. The chapter's single move is that you never solve it with one test. You compose a cascade of filters, each keyed to a different observable, each narrowing the population before handing it to the next. The 2 coarsest are named up front. Selection kills every cell lacking the trait, that is, survival under antibiotic pressure. Screening lets all survive and interrogates each colony individually. Downstream filters grow more specific. Hybridization uses sequence complementarity itself as the address. Antibodies target the expressed protein. Identification is thus successive sieving. The population collapses to one because each stage exploits a property the target has and the background does not. This section explains what changes when the object of study shifts from the single gene to the whole genome. The chapter pivots the book away from how to cut and clone genes toward what that machinery buys as pure science. The answer is a change of unit. The object of explanation rescales from the isolated gene to the whole genome, and that rescaling is what the word genomics names. Function is relational. It is never read straight off the sequence. A gene means what it does in context, so computation can only infer that meaning, and experiment must confirm it. Reading a genome at that scale forces nested resolution. You move through the genetic map, then the physical map, then the clone map, and finally the sequence. Each one is a landmark grid, finer than the last, the arrangement the author draws as a roadmap. The same upward move regenerates from there. It produces the proteome, and it produces comparative genomics. The gene was only ever the starting point. This part explains how the cloning toolkit turns inward from a destination into an instrument. Genetic engineering here stops being the goal. The chapter opens. Part 3 by aiming the tools at a new question: what genes are and how they work. The unifying move is a single one. Every method reads an invisible functional feature by forcing it to leave a physical difference against a control. A bound protein shields its DNA from DNAs, and the gap in the latter is the binding site. That is footprinting. A protein-bound fragment drags in the gel, and that is retardation. Function is never seen directly. It is inferred from what a perturbation protects, slows, or abolishes. That single readout logic scales from one promoter up to whole genomes. This section explains how genetic engineering turns a cloned gene into a manufactured product, and what actually decides whether that product succeeds. A cloned gene is a blueprint. This chapter is where the blueprint becomes a factory, and the factory's yield is set by a single trade-off. Every expression host sits on one axis, cheap and robust at one end, faithful at the other. E. coli grows fast and costs nothing, but it cannot glycosylate. Climb toward yeast, then baculovirus, then mammalian cells, and you buy post-translational authenticity at rising cost and rising fragility. The pattern generalizes past proteins. Tennis. The chapter's deeper move is this: the science being solved does not gate success. The BST story shows the binding constraint migrating downstream to regulatory approval and market acceptance, where a technically finished product can still die. This section explains what happens after a gene is cloned, when the book turns from technique to production. A cloned gene is not yet a product. This chapter is where the book's toolkit turns outward and asks what it manufactures. The generative move is simple. Biotechnology already means using an organism or an enzyme as a production instrument, and gene manipulation merely swaps in a modified instrument. Nothing conceptually new is added, only a better tool instrument. From that one move, everything downstream follows. Expression demands two co-optimized layers. The first is the biology, the host, the promoter, and the choice between a native protein and a fusion. The second is the process, scale-up, and purification. The chapter's understated surprise is carried by the BST anchor. The science is the tractable part. The binding constraint migrates to regulation and to market acceptance. Chymosin, recombinant BST, and insulin are one pattern seen 3 times. This section explains the single move behind the whole chapter on medical and forensic applications of gene manipulation. The whole chapter runs on one move. Once you can read and write DNA, medicine, and law, stop needing proxies. The nucleotide sequence itself becomes the thing you observe, so you interrogate the cause directly rather than its downstream shadow. Diagnosis reads the causal sequence directly. The HIV probe assays viral DNA inside the T lymphocytes, rather than waiting weeks for anti-HIV antibodies to appear, so the false negative window collapses. The same logic detects a mutant allele, or the genetic signature of a pathogen. Therapy writes the sequence back. You deliver a corrected transgene into the patient, as in AADA, deficiency, and cystic fibrosis. Forensics reads identity of hypervariable loci, displacing the older reliance on blood type. 3 applications, 1 primitive. Because the nucleotide sequence is the observable, you question the cause itself instead of its downstream shadows, antibody titra, phenotype, serology. This section shows how one capability generates the whole chapter. Once you can ask of any sample whether this exact sequence is present and whose it is, everything else follows. Every application here is that single read operation pointed at a different question. Medical diagnosis asks whether a pathogenic allele or viral DNA sits inside the patient. HIV is detected in T lymphocytes before antibodies exist, and the false negative window closes because you read the virus directly. Forensics asks whose sequence a trace belongs to. The same probe. And PCR primitive, 2 questions. Gene therapy is the inversion. It writes where the rest of the chapter reads. This is where the book's arc turns inward. The tools built earlier in chapters 4 through 8 and the genome mapped in chapter 9 now point at the human being. Reading a sequence becomes reading a person. This section explains what makes an organism transgenic and why that single distinction breaks an old barrier. A transgenic organism is defined by where the change lands. It lands in the germline, so the alteration propagates through reproduction instead of dying with the individual. That is the one line separating it from somatic gene therapy, whose transgene stays trapped in a single body. That shift dissolves the oldest constraint on recombination. Classical breeding crosses only sexually compatible partners, which locks traits inside a species. A vector that inserts chosen DNA straight into the genome erases the compatibility requirement entirely. The chapter's compression is that the recombinant toolkit built for microbes now reaches whole plants and animals. In plants, it is carried by a hijacked natural transferer, the T plasmid of Agrobacterium, which already smuggles its tDNA into the host genome and integrates it heritably. This part explains what actually defines a transgenic organism and why Chapter 12 is really one problem in disguise. A transgenic organism is defined not by which gene you add, but by where it lands, the germline. So the change propagates by reproduction rather than dying with the individual. That is the exact hinge separating it from somatic gene therapy. Chapter 12's whole thesis is one problem worn in 2 costumes. Deliver a defined sequence into a cell early enough that every descendant cell, including the gametes, inherits it. Plants and animals differ only in the delivery vehicle. In plants, Agrobacterium's T plasmid smuggles its tDNA into the plant genome. In mice, you use pronuclear microinjection, or ES. Once you see germline integration in a single early cell as the invariant, every technique in the chapter becomes a species-specific answer to it. This section explains the second meaning that the word cloning carries. Everywhere else in this book, cloning meant copying a sequence. Isolate a gene, amplify it. Here, the unit jumps scale. Cloning now means copying the whole organism from a single body cell. What licenses that jump is one primitive nuclear totipotency. Every differentiated cell keeps the complete generative program, not a diminished remnant. Weissman guessed the genome erodes with each division. Embryo splitting and nuclear transfer refuted him. Differentiation is silencing, not loss. So cloning is running that intact program backwards, deprogramming an adult nucleus to the zygotic state and letting development replay. Dolly is the proof that the silence is reversible. The information was never spent. This section explains the 13th concept, the body, and why one word in the book names two orthogonal operations. Everywhere else, the book clones a gene, meaning it extracts one sequence from the genome. Here, the same word clones an organism, meaning it regenerates a whole individual from a single cell. The primitive that links the two is nuclear totipotency. Every somatic nucleus keeps the complete instruction set, so differentiation is a reversible switch, and the full information is retained rather than discarded. Development is therefore deprogramming clay backwards. The only real question was ever, how far back a specialized nucleus can be reset. Dolly is the existence proof that the reset reaches all the way. An adult, terminally differentiated mammary nucleus was coaxed to G0 and dropped into an enucleated egg, and it rebuilt an entire sheep. The genome was never spent. It was only silenced. This part explains why genetic engineering carries no moral sign of its own. The valence lives entirely in the coupling between a capability and whoever deploys it. Nicholl closes the book by dissolving its animating question, is this good or bad, as malformed? Neutrality is a property of the process. Moral charge is a property of the application. The two only look separable. The conserved quantity is responsibility, and the chapter's real surprise is where it leaks. The instant science hardens into commercial technology, individual integrity no longer bounds the outcome, and corporate incentive redistributes the accountability. The titles, Spliced Monsters, Frankenstein, Inside Pandora's Box, enact the very move they name. They join what nature keeps apart. This section explains how the book ends by turning against its own title. The book closes by refusing that title. The question, is it good or bad, has no answer. The reason is not that the question is hard. Moral weight lives in the application and in the wielder, never in the technique, which is inert by itself. Consider the often quoted move, the atomic bomb, neutral physics, non-neutral use. The book declares this an artificial distinction. Process and application cannot be cleanly severed once a society has been shaped by the technology. The closing splice, Frankenstein's monster inside Pandora's box, enacts the very operation the whole book taught. It joins two things that nature keeps apart, to make something that would not otherwise exist. Responsibility, then, distributes to those who discover, adapt, and regulate. The debate itself is the only real deliverable.